So thank you for coming tonight, or I guess staying in your house tonight. Thank you for doing that during a pandemic. What we'll talk about tonight is a little bit about some stressors that pollinators are experiencing. So I'm sure some of you are very familiar with these, but if you're not, we'll talk a little bit about those stressors. We'll talk about some of the research that we're doing on 3 of those stressors. We can't cover everything tonight, but we'll cover 3 of them. I catered the talk to try and focus the most on plants as possible. So maybe some new information about plants, especially native plants that you guys might be interested in. And then some things that you can actually do. So this is a major part of my mission, or our mission as this lab, is not trying to just research what these stressors are and how they're impacting pollinators, but try and come up with some management decisions based off of what we're actually finding to say, okay, well, how can we actually help our pollinators, especially here in New York, but obviously some of or hopefully most, but at least some of the take-homes here are applicable beyond New York as well. All right, so before I get started, I'm going to just thank everyone, just in case I don't have time to thank everyone at the end here. Anytime I say I during this talk, I actually mean them who did all of this work. So I have a wonderful lab. Some people to point out here are postdoc Aaron Iverson, who I'll talk about some of the pesticide research, but he's also a wonderful botanist, the best plant ID person who I've ever worked with. He pretty much can ID anything on the spot, really impressive. But we got him into bees for a bit. And then Nico Baert, so he's our pesticide chemist, takes pride in knowing nothing about bees, a little bit about plants, and a lot about chemistry. Laura Figueroa is a former PhD student who I'll show some of the parasite transmission work at flowers. Maria Van Dyke, who I believe is actually a member of this group, she is a technician split between my lab and Brian Danforth's lab, and producing 2 of maybe the key extension publications that you guys might be interested in. So really doing some great work with native bee systematics, but also she just is wonderful at putting out lots of very useful information for folks. Paige Muniz is a technician in my lab. Well, she's actually my lab manager now. She's also a native bee systematics expert, so she can ID all of those bees that you say, looks like something small and buzzing around on my flowers. Well, she's really good at IDing all of those. Ashley Firsch is a former technician in the lab. Emma Walters is our honeybee extension associate here at Cornell. So we'll talk a little bit about the New York State Beekeeper Tech Team. And then Travis Grout, final person to highlight here. He's our agricultural economist on the New York State Beekeeper Tech Team. So not a biologist, but dealing a lot with some of the economic repercussions that certainly tie into biology of pollinator health. But he also took lead on writing our, I guess, apparently controversial, but I think just mostly a tome of information. We wrote a 432-page assessment for a risk assessment for neonicotinoids, neonicotinoid insecticides here in New York. So really impressive work that he did, and then all of these folks have done. Okay, so after saying thank you to them, we'll jump in. All right, so why are pollinators important? And why are bees important? Well, I think you can pretty much sum it up in this picture. So bees are messy, and that means that they're really important for transferring pollen. They can transfer pollen from a male flower or flower part to a female ovary, and thus facilitate pollination and the functioning of a large portion of our world's ecosystems. It's also worth a lot of money, what they do. So here are some of the major pollination-dependent crops that we have in New York. We're called the Big Apple State for a reason. So you can see up in the upper left-hand corner, apples last year were worth about $320 million in New York. Soybean is also produced here in New York to a fairly large extent, somewhat reliant on pollinators. And then there's a lot of other fruits and vegetables here that are reliant on pollinators. So what we've come up with just this last year is that an estimate for how much pollinators are worth in terms of their services for agriculture here in New York. And this is based off of, for example, apples being almost 100% reliant on pollinators and especially bees for producing successful fruit. So therefore, a good chunk of that $320 million is really dependent on pollinators, whereas soybean, for example, is very little dependence on pollinators. So somewhere around 5 or 10% of an increase in yield if pollinators are involved in soybean production. They're a good selfer. And then some of these other fruits and vegetables, to varying extents rely on pollinators. So we estimate that they contribute about $400 million in pollination services annually in New York. Just to put this in a broader perspective, you guys have probably heard that around 3/4 of the world's crops rely on pollination. If you haven't, here's the reference. And we estimate, at least as of several years ago now, we need a better updated reference on this, but at least as in terms of around 10 years or so ago, we estimate that about $170 billion per year is attributed to pollination. So $170 billion per year to the world's economy is attributed to pollination. Here in New York, in addition to all those pollination services that pollinators and bees especially provide, we also have a large beekeeping industry. So we have about 3,000 beekeepers here in New York. Maybe some of you on this call, maybe not. All of those beekeepers keep somewhere around 100,000 colonies at any given time here in New York State, and on average are producing about 4 million pounds of honey each year. So that's worth another $10 million on top of the around $400 million that pollination services are contributing to the state's economy. So that's on average. Here's a situation over the past 30 years. So since 1987, since we've been systematically taking data on honey production in New York State, you can see that there's a pretty clear trend for how honey production is going in the state. And in fact, when I started here at Cornell 4 years ago, the previous 13 years have been the worst honey producing years in New York State's recorded history. So there's lots of reasons for why this is occurring, but one of them is shown right here. So this is a map that was produced by the Bee Informed Partnership. That's our major national effort for recording how honeybees are doing, how their colonies are doing, and some of the stresses that are associated with them. And what you can see is that between 40 and 68% of New York honeybee colonies have died each year since data started to be gathered on this in 2006. All right. Last year here in the state, we had 100,000 colonies, and 50% of those, so 50,000 of those 100,000 colonies died. And that's obviously contributing to some of the problems that beekeepers are having. Oh, not sure why that's in there. In addition to the problems that beekeepers are having, we also know that native pollinators are not doing so well. So New York is home to 414 species of wild bees. That's our current estimate for how many different species of wild bees are out there. And we know that at least 53 of those species, or 13% of them, are experiencing some form of population declines or range contractions. That is likely an underestimate, simply because we don't have great data on a lot of these bees that just aren't very well studied. So it's a sort of lower limit for the number of species that are having problems is 13%. It's likely much greater. We also know that it's not just bees that are having problems. So here are some data that were published last year showing pollinator declines in the UK. Some data sets like this exist for the US as well, but this is just a really nice figure, so I put it in here, showing that there's been a decline, certainly in bees, in the UK over the last maybe 20 or 30 years or so. But if anything, hoverflies have been declining in their ranges even more than bees. All right, so hoverflies are also important pollinators. They just happen not to be bees. And then finally, what we've noticed over the past couple years especially is that it's not just pollinators that are declining. It's actually general insects in general, insect declines that are occurring. So here is the most recent meta-analysis on this particular phenomenon. So this was published this year, doing a worldwide meta-analysis of all the data sets that exist to date that have really shown long-term trends in insect abundance, both in a terrestrial and aquatic system. So I'm showing you terrestrial right here. So each one of these points in color show whether those long-term data sets either show a positive effect, so whether insects are increasing in blue, or whether they're decreasing in red. And I think the main take home here is twofold. So number one, you can see that it's not declines everywhere, right? So there is some heterogeneity for what's going on. Some places you actually see increases in abundance, and other places you see decreases, and others you don't see much. So I think that's an important take-home message. But the other take-home message that's perhaps even more important is when you sum all of those up, and when you look at the overall global trends, we see a decline of 9% per decade in insect abundance. That means nearly a third of our insects have been lost over the last 30 years. That is a staggering amount of loss of terrestrial insect abundance. Okay, so why are pollinators doing so poorly? And you can extend a lot of these stresses that I'm going to show you here just to general insects, not necessarily all of these, but certainly if we want to focus just on pollinators for this talk, we'll stick to 5 categories. And these are 5 categories that have really come into to their own as sort of being the scientific consensus for why pollinators are experiencing declines. So the first is pesticides. It's agrochemicals. That includes insecticides, fungicides, and herbicides. We'll talk a little bit about those in this talk. The second is pests and pathogens. So especially novel pests and pathogens, such as the varroa mite that's shown as that big sort of maroon dinner plate looking thing up in the upper left-hand corner here. So that mite made a host switch from Apis cerana, the eastern honeybee, onto Apis mellifera, the western honeybee, which is shown just to the side of the pests and pathogens, a little decal here. And it turns out that, well, the western honeybee doesn't have the co-evolved defense mechanisms against that varroa mite, and therefore is very susceptible not only to the mite, but also the viruses that it vectors, such as deformed wing virus, as you can see is being expressed in that particular bee right there. We also know that climate change is having a major impact on pollinators. So it's influencing things like phenological mismatch. So the plants are queuing into slightly different cues. You know, for example, early in the spring, we have a great data set from Geneva showing that apple bloom is getting earlier and earlier every single year. That's also true for our native plants and other species of plants. Even the invasives are changing when they're actually coming out in bloom, especially in the spring. It turns out that some of the cues the plants are keying into are not the same things that the pollinators are keying into. So if you have a plant coming out a week earlier now, and a pollinator that's keying into slightly different things, but specializes on that plant and a few other things, it's sometimes mismatched. So it's coming out at different times, or it's emerging at different times than a lot of its food sources. And then also droughts, you know, contributing to things like fires that we're seeing in the American West right now. Obviously, those massive fires are having big influences on pollinator communities, in addition to pretty much every other community that's going on out there, including plants, and things like rain and floods here in the Northeast or the Southeast as well. Okay, the fourth main factor that's influencing pollinators is habitat. So either a low diversity habitat or lack of floral resources. So low diversity could happen in, you know, like the picture I'm showing you right here of a monoculture crop such as corn. Corn certainly blooms at one point in the year, and bees do, you know, use that pollen. It's a wind pollinated plant, but it's still a protein source for bees. So bees will use it, but it's only blooming at that one time of year. So if you have an entire landscape that's covered in corn, or maybe corn and soybean, there's very few floral resources for the vast majority of the year. And then finally, for managed bees especially the western honeybee, which is our main managed bee in the world, if you have inadequate management practices, so if you don't treat your bees very well, that's obviously bad for them. So I put this picture up here for a reason. So this is an 18-wheeler getting a whole bunch of honeybee colonies in New York ready to go out to California for almond pollination. And that occurs in February every single year. So at the end of January or early February, we have about 60% of our honeybee colonies in the state make the trip across the country to pollinate California almond. Turns out that's true for the rest of the country as well. So somewhere around 60% of all the honeybee colonies in the United States make that trip to California. It is, in fact, the largest insect migration that occurs in the world. It's assisted migration. It's not the monarchs that are doing it on their own and going down to Mexico. It's on the back of 18-wheelers. But it turns out when you do that migration in the middle of winter, it can be stressful on those pollinators. Okay. All right, so if you remember nothing else from this talk, just remember this slide. So these are all the major stressors, that are contributing to pollinator declines. We're going to touch on 3 of them today, and well, tonight. And we're not going to touch on any of them in great detail, but I'm going to hopefully give you a little taste of some of the stuff that we're doing and some of the actions that you can potentially take to help pollinators in terms of habitat, reducing pesticide exposure, and reducing parasite transmission. All right, so we'll start with habitat. And I should say, I don't know how many people are on this call, but any questions, before we jump into things? I think it's a relatively small group, so if anyone has any questions now, that's totally fine, or I can keep going. All right, I'll keep going, but please jump in if you have any questions. All right, so we'll start with habitat. All right, so I'm going to tell you one story about habitat here, which is directly relevant to where we live. So when I started out here at Cornell, I had a master's student, Nelson Milano, and he was very interested in figuring out which habitats bees thrive in here in New York. So what he did is he put these sentinel bumblebee colonies out in 4 different, major habitat types, and he put these well replicated in all of the locations that you can see here. So up by Rochester, Geneva, Syracuse, and down by Ithaca, either in conventional agricultural settings, organic agricultural settings, natural areas, so sort of a mix of woods and fields, and then suburban areas. Really to ask the question, well, how would these bumblebee colonies perform in each of these areas, so we can get a sense for, well, which are the areas where we should really prioritize in terms of doing something for potentially improving the situation. All right, so he put 64 colonies out in each of 2 years, so it was 128 colonies that he put out and monitored over the course of 2 years. Really impressive work. And we can summarize the main finding in one figure. So here is his figure showing colony weight gain in each of these 4 habitats. And what you can see is the bees did fairly well in organic agricultural areas, natural areas, and conventional agricultural areas. In fact, there was no difference in the weight gain that they had, so that as a metric of how they performed. So bumblebee colonies, if they perform really well, they're going to gain a lot of weight, produce lots of sexual offspring at the end of the season, which is directly proportional to how much weight they've gained, and that's their fitness. But you can see in suburban areas, we had about a 40% reduction in colony weight. So the bees were not doing well in suburban areas. So we looked into a lot of different things. So we measured pesticide residues, we looked at parasites in these colonies, we looked at a lot of different factors that could potentially be explaining this. And in Nelson's original data set, what we found is that the best predictor of colony weight gain, so this is relative colony growth here, was the type or the amount of development in those areas. So if you had highly developed areas with a lot of impervious surface, so basically just a lot of asphalt and concrete, the bees didn't do all that well. What we also did is we teamed up with Aaron Iverson. So he's our native plant ecologist in the lab. And what he had developed before joining our lab was a really impressive map of floral resources in this general region. So he had 2 years of plant identification and land cover surveys in upstate New York. So he surveyed 462 different plant species at 20 different land cover types. And then he counted all the flowers on each of these plant species and came up with an average for each species for how much flowers was really produced in each of these land cover types. With the idea here that, if you integrate in this circle here, let's say a bumblebee, your average bumblebee in a bumblebee colony is going to have a foraging radius of about 1.5 kilometers around that colony. Well, if you know that colony's location, on average how many flowers and at what time of year those flowers are going to be available for that actual colony. All right. So here are some actual data from this model that Aaron created, and I think it's actually a really great representation of what we see in terms of bloom here in upstate New York. So across all these different land cover types shown by the different types of or different line colors here, you can see that yes, late May and early June, you have this really big flush of trees that are in bloom, trees and shrubs like black locust, American basswood, cherries, etc. So that's contributing a lot to the flowers that are available. Then it dies off for a little bit. And then you start to see a trickle of some of these roadside ditches and some of the disturbed areas, other things like old fields where you have chicory, thistles, clovers, parsnips, etc. in July. Then again, there's a little bit of a drop off. And then the old fields really kick in with the asters. So goldenrods and asters, the stuff that we just saw. So you really see these sort of 3 humps throughout the season. And I would say that's pretty, it matches what I see when I go and drive around upstate New York. All right, so if we use that model and look across all of our different habitat types for where Nelson had his bees, what you can see is that we found fewer floral resources in these suburban areas as well. So it wasn't just impervious surfaces like concrete and asphalt in these suburban areas where most of us actually live, but it turns out that there's fewer floral resources here as well. All right, and this is just on the y-axis here. This is peak floral area for that time that we actually had the bumblebees out in these locations from June to September. I would say that, you could intuitively even say that this was going to be the result. So if I think of suburbia, I think of this. Maybe not necessarily in Ithaca, but at a lot of other places that I see suburbia, it really is this, you get this sort of picture. So people just love their lawns. The parks that people have either in Central Park here or in more of an upstate New York park, they're filled with these green spaces. And green spaces is really, that's it, even in the vernacular. This is thought of as a positive thing, as a green space. There's a positive association with that particular term. So I guess my question is, why are green lawns beautiful? Why are these green spaces beautiful? Why not pink, purple, and yellow lawns, things that are filled with flowers, if we now have some good evidence that those particular spaces could really help pollinators? So if pollinators are not doing well in these suburban areas, what can we do? How can we sort of increase, or how can we get people to, appreciate more flowers in these areas? Well, I think there's 3 ways to do this. And I'm going to show you the first one here. So let's say you want your lawn, whether well, I guess this is the Finger Lakes Native Plant Society. So maybe you guys don't want your lawn, but maybe your neighbors want your lawn. So if your neighbors want their lawns, maybe they can have their cake and eat it too. So if you have a lawn, why not create a bee lawn? So this is a wonderful project that has been developed over the past several years at University of Minnesota, where you can go to their website, just Google bee lawns. And what they have done, especially in the Minneapolis area, especially where the rusty patch bumblebee has been a major conservation concern, they've gotten a lot of folks to start to create these bee lawns. And what is a bee lawn? Oh, sorry. Well, before I get there, they've even got the Minnesota State Legislature interested in this. So the Minnesota State Legislature has, in 2019, so last year, they actually committed nearly $1 million in incentives to get homeowners in, especially the Minneapolis area, incentives to create these bee lawns and get more flowers in their turfgrass area. So what is a bee lawn? This is a picture from just this afternoon. This is my mix that I bought from one of these companies. There's several companies that sell this bee lawn mix. And it's grass, but it's also a lot of these small flowers. So low-lying flowers. Let's see if I can go, whoops. Low-lying flowers, just like these clovers, some different lotus species. Some of these things where you can still have a lawn, but you can also create floral resources. All right. Okay. The second thing I think you can do is you can make a pollinator-friendly garden. Okay. So let's say you want your lawn, or you don't have a lawn, but you really want to focus on improving your garden. So we have a wonderful new resource that was created by Maria Van Dyke, and this is now available. It's not quite up on our website yet. I'm getting it up, I think, in the next couple of days. I have to talk to Maria about this. But she's developed this booklet. It's a 32-page booklet. It's basically a how-to guide for choosing native plant species that specialist native bees, which are oftentimes the ones that are rare or of conservation concern, choosing those plant species that will really be attractive to these particular species. Okay, this is a wonderful new booklet. We will get it up very soon, and I'm happy, even before then, I'm happy to send it around to anyone who's interested. And then finally, some things that we're doing that you guys may be interested in. Obviously, you may not have a whole solar, power station in your backyard, but you've probably seen some of these popping up around here. So one just down the street from me here on Turkey Hill Road, we're working on, and then we're working on some from around the state as well, where there's a lot of interest in not just having turf grass under these solar panels, but actually doing something else with that land. And in fact, this is a lot of land in New York that's going to become used as solar, utility scale solar facilities. So Governor Cuomo has put this, goal of having 40,000 acres of land used as solar power. So what we're doing right now is we're working with a company and a few other folks to try and figure out which seed mixes actually establish best. So can we make pollinator-friendly solar plantings? And then we're trying to assess, well, it's not just, it is, I guess, an assumption that you are making an impact by having these seed mixes and creating lots of habitat for pollinators, but no one's actually shown that this really is having an impact. So what we're trying to do now is, assess bee diversity and abundance over time to make sure that, it's not just a pretty picture, but this really is benefiting pollinators. Okay, so there's some things that you can do for habitat or some projects that we're involved in. Now we'll talk a little bit about pesticides, and then we'll finish up with pests and pathogens. All right. So when I first started here, again, a lot of the beekeepers were telling me that when they went in and did apple pollination, that the pesticides were killing the bees. So they were having really a hard time after, coming out of pollination, that, that the bees were not doing well and they thought it was pesticides. So I said, okay, well, that's one possibility. It could be lots of other things. It could be the fact that they're only eating apple pollen. It could be the fact that, well, the landscapes where apples are just generally harsh. Maybe there's water issues or other problems during pollination. So I said, well, if we really want to pin this down on pesticides, let's actually look at the risk from pesticides, which, believe it or not, hadn't been done up until a few years ago. So what we did is we as a lab that works with honeybees, what we did is we mimicked commercial pollination at 30 different apple orchards here in New York. You can see all the different locations there in red dots. And yeah, a cartoon of what this looked like. We put 120 experimental hives into 30 orchards, 4 hives into each orchard, and then measured pesticide exposure. We gathered pollen, from the hives, and then we brought it into our lab. Here's Nico in our lab with our Lamborghini. So this is our LCMS, where we measure lots of different things, including pesticides. So we have a 260 compound multi-residue pesticide analysis assay that we can look for basically anything that's sprayed out there. And I'm going to summarize all of the results in one slide. I could give a whole talk on just this study, but we're going to summarize everything in one slide. So what you're looking at here is all of the different 30 orchards across the x-axis. So just anonymized as a letter. So we're not putting names on anyone. And then on the y-axis, we're looking at the pollen hazard quotient. So that is expressed as a percentage of a contact LD50. So if you're at 100% of an LD50, that means, on average, 50% of those bees are dying acutely from pesticide exposure. And what a pollen hazard quotient is essentially saying, of all of the different pesticides in there, we know some are really toxic at each of the sites. We know some are really not toxic, and then we know how much of each pesticide was actually in there. So in each of those bars in this graph, you've got a lot of different pesticides and a lot of different toxicities, basically. Okay, so the take home from this is it's a mixed bag. So sometimes, yeah, there is high risk from pesticides. At 2 of the 30 orchards, we're above this blue line cutoff. You can see my cursor here. So this is the US EPA acute exposure level of concern. At 5 of the orchards, we're above the European Food Safety acute exposure level of concern. So this is just a more conservative estimate that Europe makes compared to the US. And then at 22 of the orchards, so 22 of these all the way over to here, we're above the European Food Safety Authority chronic exposure level of concern for 10-day exposure. And we had these bees in there for 10 days, so we could actually measure this. So I would say the major picture here is, yeah, it's heterogeneous. So some orchards on the right-hand side here are doing fairly well, but a lot of orchards are not doing well. So we actually know that pollen from 22 out of those 30 orchards is above a regulatory agency level of concern. All right, so there's, maybe this high risk event that beekeepers are going to do in New York for pollinating apple. What about normal day-to-day pesticide risk? So a lot of beekeepers ask me, or just a lot of citizens ask me, what is risk to bees just in a normal day-to-day context? So here, I'm going to show you some data from when we started this New York State Beekeeper Tech team in 2016. The results are almost always the same each year. We've been doing this for the past 4 years, but I'm just going to show you the first year's data. So what we did that first year, we went out and sampled 198 colonies from across New York. You can see us sampling some of the wax here and putting it into vials. And here's a histogram showing the results. So out of those 198 colonies that we sampled wax from, on average, we found 5.5 pesticides per colony. Okay, so this is the number of pesticides in each colony on the x-axis, and then the number of colonies on the y-axis. One colony had 18 different pesticides in the wax, and only one colony had no pesticides. All right, so it didn't matter whether you're a hobby beekeeper, or sort of a small scale person, sideliner, or commercial beekeeper, someone who's got over 500 colonies and is really making your living on this. You really did find a lot of pesticides in your colonies. All right, so pesticides are ubiquitous. Bees are basically always exposed to them, at least honeybees are. But what about risk? Okay, so now we're going to go back to this hazard quotient, where it weights not just what pesticides are there, but what quantities they're there in and how toxic each of those pesticides are. What I'm showing you here is the majority of the commercial beekeepers that we sampled here. So this was a number of those commercial beekeepers, and results were very similar for the sideliners and also the hobbyist beekeepers. So now what you can see is we're not upwards of 60, 40, 60, 80% of a contact LD50. Now we're down below 1% of a contact LD50. So I would say this is a fundamentally different conclusion from apple bloom. When beekeepers or native bees are exposed to these pesticides during apple pollination, yeah, there's some risk. But when it's just day to day, going about doing their business, yeah, there's pesticides there, but it's very low risk, okay? But, so when we actually look at the particular pesticides that are there, We know that these are their top 13. So this was the top 13 pesticides we found in 2016, and it's relatively similar every time we've looked since then as well. So our top pesticide that we found was piperonyl butoxide. That's always comes back as our top one. It's present almost everywhere. I think if I dug up some soil from my yard, I would probably find piperonyl butoxide. It just seems to be everywhere. I'm not sure why. So we found that there were 196 positive detections out of 198 samples. So a lot of piperonyl butoxide out there, and then a lot of this purple stuff. So what is piperonyl butoxide and what is the purple stuff? Piperonyl butoxide is a synergist. So what that is it is a compound that causes insecticides to become more toxic. If you look at the can of wasp spray that you might have in your basement, oftentimes what it has is pyrethroids, so pyrethrins or something along those lines, and piperonyl butoxide in there. That is what kills those wasps so quickly, is the combination of the insecticide and the synergist that is causing that insecticide to become acutely or more acutely toxic to that insect. In addition, a lot of these fungicides, which are shown in purple here, we are finding out have synergistic capacity as well. So they're not meant to be synergists, but they just happen to synergize with insecticides. In fact, of those 198 samples, the wax samples that we took, we know that 163 of them, so about a little over 80%, contain pesticides combinations known to be synergistic. That is important because when we're computing hazard quotients, we do not assume that any of these are synergistic. We assume everything is additive. So we know because of the presence of these synergists that we are underestimating pesticide risk to bees. So just a picture to show what a synergism actually looks like. So this is Aaron really getting into his bee research here. We stole him from the plant world and made him a bee toxicologist for a year. Now he's back to plants again. But this was one really nice paper a really nice study that he published last year showing what a synergism looks like. So you can see here, if we feed bees sucrose in green here, they do great. And well, that's no surprise, right? So if a bee is consuming sucrose, it should like it. And 96 hours after we feed them sucrose, they should survive just fine. This is probability of survival on the y-axis here. If we feed them a fungicide, myclobutanol in this case, the bees do just fine. So fungicide is very non-toxic on its own to bees. If you feed them bifenthrin, a pyrethroid insecticide, at levels that they would be typically exposed to in apple bloom, for example, which is what we base these concentrations off of, you can see that some proportion of those bees will die. So somewhere around 30% or so will die over 96 hours. But if you feed them both bifenthrin and myclobutanol, now you're really killing bees. So this is a synergism, what we're seeing right here. That's what this is noted by the stars. That means it's a non-additive response. So this fungicide is interacting with that insecticide and really causing things to become toxic. All right. We think this is one of the major reasons why folks like ourselves and other labs are finding these types of patterns. So here was a study that we published a few years ago now, looking at the best predictors of pollinator, or these declines of these four bumblebee species across the United States. So you can see based off of some historical population data and then a present day sampling that occurred in 2009 in this case. We were expecting to find these bumblebees in all of the sites where there's a point, but we only found them in the places where there was black. So of all the places that were in red that we did not find them, we asked, well, what are the best landscape predictors? So this is one of these new machine learning data sets where we sort of threw the kitchen sink of predictors in here. We said, what about habitat change? What about urbanization? What about, weather? Things like climate change, temperature differences, and then also a whole bunch of other predictors with pesticides. And what we found is the best predictor of loss of these species from those sites was fungicides, which you can see in this figure right here. So if you have more fungicides, you're more likely to be lost from those locations. And the fungicide usage across the US here is shown in blue overlaid on this map. All right. Okay, so doom and gloom. What can we actually do about pesticides? So one thing that we can do is, have regulation step in. So this is, I did not ask for this, but the EU just banned the most used fungicide on the planet, chlorothalonil. Some of our analyses that I didn't show you just with this last bumblebee paper is that chlorothalonil really was the best predictor of not only declines, but also disease in these bees. And the EU used our study and some other studies as evidence to say that we need to get rid of chlorothalonil, and they just did it, which is surprising to me, but they did it. Maybe a more pragmatic approach for your everyday citizen, including myself, is to use pesticides a bit more safely. All right, so if you've got to use pesticides, especially if you're a grower of crops and you need to control your insect pests, or whether you need to control them in your garden, etc., maybe you can choose pesticides and use them a little bit better. So choose particular pesticides that maybe target your pest or plant pathogen, but don't have as severe of consequences on pollinators or other non-target organisms. So again, this is the second guide. So Maria I've already shown you the guide for prioritizing native bee species through native plant species, which I think a lot of you might be interested in. I know I am. This is the second guide that I think a lot of folks might be interested in, and these are available at the website below now. So these pesticide decision-making guides. All right, and then finally, I just wanted to put this on your radars. If you are interested in this topic of neonicotinoids, which has been in the news a lot for the past decade or so, the EU has actually banned neonicotinoids outright. Various states in the US are considering either partial bans or some form of restrictions, and that includes New York. We were commissioned by the governor's office and also a few state agencies here to really synthesize the literature on this topic. So I won't go, that would be a talk in itself. I won't go into this in any given detail, but I will say that if you're interested, this is open to the public. So you can go to that particular URL and download this report if you're interested. All right, so that was pesticides. And what I'll finish up here with is I think some of the more fun stuff that we're doing in the lab, and that's looking at parasite transmission with a take-home message that I think everyone hopefully can get around for how to reduce parasite transmission for pollinators. All right, so we're going to ask two questions for this final part of the talk here. So first, how do bees transmit disease? And then second, how does disease spread in plant pollinator communities? All right, so we'll show you a cartoon first. So if you consider this honeybee that's coming along to a flower, it might have some parasite particles, viruses or whatever other pathogen might be on there in blue. It forages at that flower and potentially leaves some of those parasite particles there. So think about this as, us all, using hand sanitizer nowadays after we touch dirty doorknobs because we're worried that COVID might be on those dirty doorknobs. Well, here is a flower, maybe the analogy to a dirty doorknob stands. I'll show you some data on that. Okay, so now a bee comes along. It has been maybe a different species of bee, and it visits that same flower. It potentially not just picks up, nectar and pollen, but also potentially picks up some parasites, goes on its way, and either does or does not become infected. All right. So do we have any evidence that this actually does occur? Yeah, we have a lot of evidence. So my lab and some other labs have been really pushing this over the last couple years to really figure out how important floral transmission of parasites are. Here's just some data from Laura Figueroa from a paper she published last year, where she tested parasite transmission of this trypanosome parasite, Crithidia bombae, of 3 different species. And what you can see here is in the figure is that it didn't matter whether it was Lobelia, Lythrum, or Monarda. Parasite transmission did occur at each of those species, but it varied by where the parasite was on those different flower parts. So whether it was inside the flower, outside the flower, or on the bract, okay? It turns out that we also have lots of data showing that it varies where different pollinators deposit parasites on flowers. So there's lots of interesting heterogeneity that's going on here, which I'm not going to get into for this talk, but we would be happy to talk about it at some other time. Okay, so that's how bees transmit disease, at least in the environment. We know that there's some really good data showing that transmission at flowers occurs. So how does disease spread actually occur in plant pollinator communities? So here we're going to go out to some old fields just around here in Ithaca, New York, where we did season-long samplings of 3 of these different old fields. And we screened for 5 different parasites, almost 3,000 individual bees from about 110 species, and almost 3,000 flowers from 89 species. The parasites that we screened for, so we had 2 microsporidians, Nosema bombi and Nosema serrani, 2 very prevalent parasites in lots of different bee species, a neogregarine, so a protozoan, an Apicystis species, and then 2 different trypanosomes, which are also highly prevalent in the community, Crithidia bombi and Crithidia expoichi. What do we find? So if we look at all of those different parasites in each of the bee species here, so now this is shown with bee genera. There's just too many bee species to put in one figure, so this is by genus. But here's a heat map showing, the amount of parasite in each of these bee genera. And what you can see is that on average, we have 42 bee species that actually have parasites, and 16 individual bee species, 16% of individual bees have parasites. All right, so we have a prevalence of all of those 3,000 bees, 16% of them have a parasite in them. What about flowers? All right, so I'd say that was not necessarily new information. We have some other data sets showing that, yeah, it's about that prevalence when we look at bees in the community. Not like the whole community is sick, but a decent proportion of them have some form of parasite. But what about flowers? What do we see? This is what blew my mind. So of all of the things that I have seen, all of the results I've seen over the past maybe 5 years or so, I think this is one of the more exciting results that has sort of fundamentally changed the way that I look at flowers. So overall, you can see all the different flower species now here, flower genera on the X or Y axis here, and then the 5 different parasites, and you can see a lot of green in there. So what we find is that 70% of flower species have bee parasites on them, and individual flowers in old fields have bee parasites on them. That means that 1 out of 11 individual flowers has a bee parasite on it. Totally wild. I would never have guessed that it was that high prevalence on flowers. Okay, so how does disease spread actually occur in these complex bee flower communities? I'm sure if you guys go out into either natural areas here or look in your garden, you'll see something like this, if you look over time. So this is a plant pollinator network. So all of the different bee species are shown here on the left. All of the different plant species and their flowers are shown on the right. Okay, so this is just connecting the dots between all of the bees that visit each type of plant species. So what happens if a honeybee gets sick? So shown here in red is highlighted all of the different plant species that visits it and potentially transmits, pathogens at that particular, flower species. Well, it turns out that it's a really short avenue. So if you have one bee species that gets sick and it visits those flower species, well, it's a pretty short, transmission event to just hit a lot of other bee species. So this is the bounce back from if you just have 1 degree of separation here. So this is now the other bee species that are visiting those same exact flowers, and then shown whether they actually do share those parasites or not in either the red circles or the black circles. Okay. So if we look at how this takes place over the course of the growing season in these old fields, what we see is a general increase in parasite prevalence through time in these old fields. So this is starting in, late May when some of the first flowers start coming out in old fields and some of the first bees. So very late May is early on, sort of over, there we go, you can see my cursor over here, late May, and then mid-September is when things are wrapping up here. And you can see that it didn't matter really which parasite you were looking at or the overall trend shown in black here, there's an increase in parasite prevalence. So just to, hammer this home again, this is really similar to what we're looking at in terms of COVID right now, right? So we want to know, how is this parasite prevalence, in this case a virus, compared to the other things that we're showing on the left here in bees. But we hope that it's, with COVID, that we're not experiencing this, just an increase in parasite prevalence. I'm a little bit worried about some of these, recent things that are going on just the last week or so here. But in general, I would say, we did not notice this with COVID, at least through the summer. There was some bumps up and down, but not just a general increase. So the bees, maybe they're not doing so great in these old fields. When we look at the flowers, we see the total opposite pattern, all right? So in most of the different parasites, we see a general decline in parasite prevalence on flowers. And if you look at the overall trend, we see that flower parasites go down. So we scratched our head about this and thought, my God, what is going on? How could there be more parasites in the bees, but fewer on flowers? You would expect these two things to be correlated, right? Well, here is what we think is going on. So in these old fields, and I think this is true if you look at pretty much any old field around here, yeah, you see some flowers early on in the season. So here's total floral abundance on the y-axis, and yeah, there's some flowers. But really they start to kick in once these asters start to bloom, right? And that's when floral abundance really goes up in these old fields. If you then look at the relationship between floral abundance and parasite prevalence on flowers, you can see that as there are more flowers, which is true later in the season, showing by color here, we do find that there are fewer parasites on those flowers as a proportion of the number of flowers that are actually out there. So it's not a great relationship. It is significant, but there's a lot of other stuff going on that we're trying to figure out. Why isn't this as tight of a relationship? Maybe there's some interesting things going on with, species-specific transmission, et cetera. But there is this negative relationship. So I think the main take home from this section, which hopefully we can all get behind, is lower parasite prevalence on flowers equals a lower likelihood of transmission. This means that maybe you have one more reason to encourage lots of flowers on your landscape. So not only are you providing forage for pollinators, creating a beautiful landscape, but you're also reducing the chances of transmitting disease to your pollinators, and therefore you're increasing their health. All right. Okay, so I'll end up with a talk that way, and hopefully we have a couple more minutes for questions if you guys have any. But I'll just finish by saying that we have a very nice extension webpage. This is not made by me, so I can say it's really nice. It's made by Emma Walters. So pollinator.cals.cornell.edu, where you can find lots of information on pollinator health and various things that we're up to. We have a Facebook page, so the DICE Lab for honeybee studies, facebook.com DICE Lab. It's mostly geared towards beekeeping, but we have a lot of just general pollinator health stuff that we advertise as well. I write a monthly column for American Bee Journal. So if any of you are beekeepers, I try to summarize a new pollinator health study every single month. I just finished this last month's article this morning, actually, but it's not just regarding honeybees. So, for example, the title of this article was "What Will Growers Use If Neonicotinoid Insecticides Are Banned?" It's a pretty important topic if we're considering making any restrictions on neonicotinoids. What are the other things that can be used? And there's a lot of other topics that I tackle each month. And then finally, just another thank you to all the folks in the lab who gave their hearts and their souls for all the data that you saw here, and some thanks to some of our funding agencies, so NIH, USDA, and some local New York funding as well. Okay, so if there's any time, and if you guys if I haven't put you to sleep, I'm happy to take any questions.